Welcome to our video lecture on topic D1.1 DNA replication. This video is both the core content as well as AHL, additional higher level content. So the first half is for everybody, the second half is for my higher level friends only. Our guiding questions though for everyone today: how is new DNA produced, and how has the knowledge of DNA replication enabled applications in biotechnology? Our objectives for today: we're going to define replication. We'll talk about the importance of complementary base pairing to replication as well as its semi-conservative nature. We're going to outline the functions of some of the enzymes that help to catalyze those reactions that bind those nucleotides together. And then we're going to talk about those biotechnology applications, PCR, gel electrophoresis, and DNA profiling. That will be the end of our SL and HL content. These last few objectives will be our additional higher level only content. We're going to review nucleotide structure, and we'll talk about the directionality of DNA replication, 5' to 3'. We're going to talk about more enzymes and their functions. We'll talk about how there is some replication that is continuous and some that is non-continuous due to leading and lagging strands, and how that leads to the formation of Okazaki fragments. And then we're going to also briefly talk about the DNA proofreading function of DNA polymerase 3. So what exactly is DNA replication? Defined, it is the production of exact copies of DNA with identical base sequences. We start with some chunk of DNA. We want to make exact copies of it. I want to have 2 strands of DNA that are exactly like the parent strand of DNA. This is so that when we go through cell division, we have exact copies of the DNA that we can put into both of those daughter cells. We want these cells to be identical at the end of mitosis so that we have all of the genetic information that we need in all of our daughter cells. Replication relies a lot on complementary base pairing. When we talked about the structure of DNA, we talked about how we have these lovely complementary pairs. Cytosine and guanine will always pair up with each other. They have those lovely structures that allow for the formation of 3 hydrogen bonds between them. A's and T's, adenines and thymines, have 2 hydrogen bonds that form between them because their structures are complementary. They have this lovely matching up. When we replicate our DNA, we take advantage of this complementary base pairing. So we're going to take this original strand of DNA. We're going to separate it. Here we have those original half strands of DNA. We use complementary base pairing to add in new nucleotides. I had a C. Here's that C. We're going to use complementary base pairing to add a G. On the other side of this guy, we had that original guanine. Here's that original guanine. We're going to add the complementary base pair, the cytosine, here. Notice that that results in 2 strands of DNA that each have the same sequence of DNA as the original strand. Complementary base pairing allows us to make this happen. This is also related to the semi-conservative nature of DNA replication. Semi means half, and conserved means saved. And when we make new DNA, we actually save half of the old DNA in each new double strand. Here's our original DNA, the template DNA that's being copied. The blue stuff is old. Notice that in each of our new double-stranded pieces of DNA, half of it is blue, half of it is old, and the other half is new. We are going to save half of the old DNA in each of those new strands. And this is why we say that replication is semi-conservative. Half of the old gets saved in the new. When we're forming new DNA, we're going to take nucleotides, little pieces of adenine, thymine, cytosine, and guanine, and we're going to bind them together to make our polymer. Polymers like DNA strands, our monomers, these nucleotides. These reactions that bind the nucleotides together to form the DNA strands are condensation reactions. That makes this definitely metabolism. We have lots of enzymes that help to catalyze the reactions that bind these nucleotides together. In us eukaryotes, the replication of DNA happens in the nucleus. We don't call the juicy stuff inside the nucleus, the cytoplasm. Instead, we refer to it as the nucleoplasm. So the nucleoplasm is really just the cytoplasm that's in the nucleus. In the nucleoplasm, we have so many of these free nucleotides floating around. Lots of adenines, thymines, cytosines, and guanines that are not yet in these crazy long strands of DNA. This high concentration allows for complementary base pairing to occur naturally, spontaneously, and then we have these enzymes that will bind those nucleotides together. This enzyme is called DNA polymerase. It is specifically DNA polymerase 3. We also, in order to be able to get those free nucleotides in here so that they can complementary base pair with our original strand, we've got to open up that double-stranded piece of DNA and make it single-stranded. Helicase is the name of that enzyme. Helicase will unzip or unwind the double helix DNA, open it up so that we have access to those base pairs on the inside so that complementary base pairing can occur. Once those complementary base pairs slide into their pair, then polymerase can come along and glue together those new pieces of DNA with condensation reactions. Again, this is semi-conservative. Notice that I've got half old and half new DNA in each of my new strands of DNA. Half old and half new DNA in each of my new DNA strands that will be identical to that original strand. This is metabolism. It is enzyme catalyzed chemical reactions. This is definitely anabolic because we're building up big polymers, DNA, from these little monomers, nucleotides. I mentioned at the start of this lecture that we humans have learned so much about DNA replication that we've been able to steal it and put it to work for us in biotechnology. One of those applications is polymerase chain reaction, or PCR for short. In PCR, what we do is we take some sample of DNA that we want to copy. Let's say that I just want to copy this chunk of DNA. This chunk of DNA we want to make a copy of. We're going to make some primers that are complementary to the beginning of that sequence of DNA that we want to copy. Here are those primers. They're complementary. We're going to also add in some free nucleotides, A's, T's, C's, and G's, adenines, thymines, cytosines, and guanines, that can then complementary base pair to the DNA sample that we start with. We have our enzyme here, polymerase, that can glue together those free nucleotides. We have a mix buffer. This just helps us to adjust the saltiness or salinity and the pH of the mixture so that the enzyme can work at its very best. We throw all these things together into a tiny baby test tube called a PCR tube, and that guy gets plopped with all its stuff into this machine called a thermal cycler. The job of the thermal cycler is to change the temperature of this mixture of stuff. The beginning temperature is quite high. I definitely drew on top of that. So the beginning temperature is quite high. We're going to have almost boiling temperatures. That is going to denature our DNA. DNA is a double strand. It's usually stuck together with those hydrogen bonds between the complementary base pairs. But when it gets super hot, 95 degrees Celsius, it wiggles and jiggles so much that the double strand separates into single strands. So here we have our denatured DNA. The double strand is now single-stranded. That opens up those base pairs and makes space for our primers to bind to the complementary sequences on that sample of DNA. We call that annealing, and it only happens when I cool things down enough so that they're not wiggling and jiggling away from each other anymore. And then the thermal cycler, this machine, will heat things back up just a little bit, now to 72 Celsius. And that's going to be just the best temperature for this enzyme polymerase to glue together those A's, T's, C's, and G's that complementary bind to our template DNA. We call that extension. All of this, these three steps, can take place in under 2 minutes. And then the thermal cycler will do it all over again. It'll go back to denaturing the DNA, except instead of having only one piece of DNA, now both of these pieces that we made in the first cycle will be denatured. And then we'll cool it off, and annealing will happen to both of those pieces. And then we can extend both of those pieces. And so we'll go from one in the beginning to 2 to, at the end of our second cycle, 4. And then every time we go through this cycle, and again, it takes just about 2 minutes per cycle, we can double the amount of DNA in our tiny baby PCR test tube. So let's say that I did 10 cycles. We're going to have 2 to the 10th power, which, hold on, let me grab my calculator, is 1024. So if I run 10 cycles, that's less than 20 minutes, I'm going to have more than 1,000 times the DNA that we started with. So this allows us to make lots and lots of copies of DNA really, really fast. What's kind of fabulous about this polymerase enzyme, we call it Taq polymerase because we stole it from a bacteria called Thermus aquaticus. That's the T and that's the AQ. So Taq polymerase we stole from a bacteria, Thermus aquaticus, that you probably guessed lives in really hot water. And so our human polymerase is going to be denatured at a 72 degrees Celsius temperature, but Taq polymerase is the happiest at that temperature. So lots of things that we humans have stolen so that we can make lots of copies of DNA, which is super cool because then we can do lots of tests with that DNA. One of the ways that we can analyze the DNA that we copy in PCR is with gel electrophoresis. In electrophoresis, what we do is we make a gel, usually composed of a substance called agarose, which is a lot like Jell-O. So you can just imagine a big block of Jell-O. We have some sample wells, some little divots in here, little holes that are just the right size for us to insert some DNA samples. We add a running buffer, basically a lot of salt water, and that allows us to complete the circuit because we're going to hook this whole thing up to some electricity. Here we have the cathode, it's negatively charged. Here we have our anode, positively charged. Super cool thing about DNA, it's a little bit negatively charged. It will be repelled by the cathode. It's going to move away from the cathode. It will be attracted to the positive side of the anode. It's going to get pulled this way and repelled from the cathode. And so we end up with this DNA migrating through the agarose gel block toward the anode side of the gel box. But because this agarose is a little bit messy, there are all kinds of fibers in here that make it hard for the DNA to move through, little pieces can move faster than big pieces. The little pieces can kind of get through pretty easily all of those fibers of the agarose. The big chunks don't move so well. Imagine these pieces are like two friends stuck together holding hands in a school hallway, and these are like 20 friends holding hands together in a school hallway. The two friends, the smaller hand-holding pieces, are going to move way faster through the hallway than this big chunk of people, 20 holding hands. Same with our DNA. Little pieces can move a whole lot faster through the gel than can our big pieces. We can then take this lovely picture showing us all these different sized fragments. I have shorter bands here and longer bands here, and then I can look at the pattern of these fragments and then match them to other patterns. We can actually take pictures of our DNA fragment samples with autoradiography. We can make some of our DNA nucleotides radioactive. We can make them emit their own radiation. And because they themselves are radioactive, auto is self, they're radioactive, they can actually develop X-ray film without us having to shoot X-rays at them because they're emitting their own X-rays, kind of, sort of. So autoradiography, DNA, when we make it radioactive, can emit the radiation to develop X-ray films, and we can take pictures of the fragments from gel electrophoresis. This is showing, oh my gosh, so many samples of DNA. And here we have 23.1 kilo bases. Remember that kilo is 1,000. So these sizes, the fragments that are here, are about 23,000 bases long. Here is only about 6.6 kilo or 6,600 bases long. And the shorter they are, the further they move. And then we have these lovely fragment patterns that we can compare. We can use gel electrophoresis in the pictures that we take with autoradiography in DNA profiling, which is also known as DNA fingerprinting. This image is showing us an example of how we could use DNA profiling in a paternity case. So we have mom and we have child, and we're trying to figure out who is the daddy. We can take DNA from mom and child and each of the potential dads, do PCR and gel electrophoresis, and then we can compare the bands. The child's bands, because all of the DNA comes from either mom or dad, need to come from matching moms and dads. So when I look at this band, I notice that mom also has it. So this chunk of DNA, child inherited from mom. And this chunk of DNA, child inherited from mom, this one also. The other three bands must have been inherited from dad. Notice that this band, dad number 1 doesn't have it. Child had to have gotten it from dad, since child couldn't have gotten it from mom. And so we can rule out dad number 1. Dads 2 and 3 are still potential dads. If I look at this band, it had to come from dad since mom doesn't have it. Could be dad number 1, except we already ruled him out. Dad number 2 doesn't have it. That rules him out. Dad number 3 does. And our winner is dad number 3. We can also use this in criminal cases to verify or exonerate suspects. This is also used in a super amazing organization called the Innocence Project. And in the Innocence Project, DNA samples are analyzed to help people who have been wrongfully imprisoned. As of November 2023, 575 people who were wrongly imprisoned have been released based on DNA evidence proving that they were not the actual criminal. And on that note, my friends, we are done with the core part of the lecture. Everything from this point forward is additional higher level content only. SL friends, keep listening if you want to, but you don't have to. You will not be assessed on this content. AHL people, my higher level friends, keep on keeping on. We're going to talk here about the directionality of replication. Remember that in biochemistry and organic chemistry, we love to count the carbons. So here is our ribose or deoxyribose sugar. Ribose has the OH. Deoxyribose would only have an H here. And we count our carbons, carbon number 1, carbon number 2, 3, 4. Carbon number 5 is up here attached to our phosphate groups. What's pretty cool about DNA structure, most of the time we only have one phosphate group for each of our nucleotides. But these guys can actually have 1, 2, 3 phosphate groups. And it's the chemical energy stored in those phosphate groups that allows polymerase to bind together our nucleotides during replication. Remember that we have pyrimidines and purines. If you don't remember our amazingly weird mnemonic device, 2-puga, Y cut 1, is how we remember that pyrimidines are cytosine, uracil, and thymine, and they have only one ring, whereas our purines are guanine and adenine, and they have 2 rings each. The important pieces to pay attention to in the directionality here, we're going to be looking a lot at carbon number 3 and carbon number 5. So we're going to focus on carbon number 3 here in the corner of our pentose sugar, corner number 5 that is attached to the phosphate group. Carbon number 5 phosphate, carbon number 3 here in the corner of our sugar. Again, we're focusing on carbon number 3 here in the corner of the pentose sugar. In this instance, it's deoxyribose, and then carbon number 5, which is next to our phosphate groups. This free nucleotide floating around in the nucleoplasm, remember that the nucleoplasm is like the cytoplasm inside the nucleus. The free nucleotides don't have just one phosphate group like they do once they get into the polymer of DNA, but instead, like I said, they start with 1, 2, 3. This guy is going to slide in here because this cytosine is complementary to that guanine, and the hydrogen bonds will attract them to each other. The enzyme DNA polymerase 3 is going to chop off those phosphate groups and use the energy that is stored in those phosphate groups to glue together the phosphate group on carbon number 5 to carbon number 3 of the sugar that comes before it. This is why we say that DNA replication happens in a 5' to 3' direction. Again, let me erase my scribbles so that we can see what's happening. Carbon number 3 is right here. Carbon number 5 is right here. We're going to take the phosphate groups of carbon number 5 and use that energy to glue the 5' end of the new nucleotide to the 3' end of the nucleotide that comes before it. 5' to 3'. 5 with the phosphate groups onto 3' of the nucleotide that is previous. This chemical bond that gets formed between this new nucleotide and the previous one is a covalent bond, a special kind of covalent bond called a phosphodiester bond. Phosphodiester bond, special kind of covalent bond between this phosphate group and this sugar. And this, again, is our sugar-phosphate backbone of DNA and also of RNA. But this example clearly is DNA. The directionality of replication, the fact that polymerase can only bind nucleotides together in a 5' to 3' direction, combined with the anti-parallel structure of DNA strands, makes things pretty complex. Remember that anti-parallel, that's an A and an N. Anti-parallel refers to the structure of DNA as running in opposite directions. So one strand of the DNA will have the 3' or deoxyribose corner end here, and then the opposite end of that strand will be the 5' phosphate end. The opposite strand will run in the opposite direction. So where I have 5' phosphate here, I have 3' carbon in deoxyribose, and then at the opposite side, we've got that 5' phosphate. The leading strand, super convenient, because this is 3 and this is 5, and because we can add 5' to 3', it's super easy for polymerase to continuously replicate along the DNA strand. Helicase, remember that helicase is the enzyme that unwinds or unzips the double helix, will open up the DNA. We call this the replication fork. And polymerase can just flow along right behind the replication fork, continuously binding together new A's, T's, C's, and G's as helicase continues to open up the DNA. On the opposite strand, the strand that we call the lagging strand, this is the 5' end, which means that this is 3', which means that we are copying the DNA in this direction. So I open up the DNA and we start here and copy that way. We open up the DNA a little bit more, we start here and copy away from the replication fork. We call this discontinuous because clearly it is not in a continuous strand. And this is due to the fact that we can only bind things together in a 5' to 3' direction. If this is 5, I cannot go 3 to 5. Instead, I have to start a new chunk here that's 3, and I can grow it in this direction from our 5 to our 3. Each of these little pieces is called an Okazaki fragment. This is the discontinuous side of DNA replication. It is the lagging strand. We are replicating the DNA away from the replication fork. A few slides ago, we talked about 2 of the enzymes that catalyze the reactions of DNA replication. Here we're going to add a few more to our list. We already talked about helicase. Helicase separates the DNA by breaking the hydrogen bonds between complementary base pairs. Interestingly, those complementary base pairs are still complementary. As soon as helicase moves away, they're going to want to be attracted to each other again and zip back up. We add in these SSB proteins, or single-strand binding proteins, that just help to hold the DNA apart. We don't let it zip back together with those SSB proteins, or single-strand binding proteins. We add in another enzyme called gyrase, which is sometimes also called topoisomerase. And its job is to relax the supercoiling of the DNA. You can imagine if we're trying to untwist some super twisty stuff really fast, we're going to get some knots formed in the DNA ahead of the replication fork. Dry erase comes in and just relaxes that supercoiling so that we don't get too many twisty knots stuck in our DNA before we can unzip it. We're going to add in this crazy enzyme called primase. And sometimes we call it DNA primase, sometimes we call it RNA primase, because it puts down a primer of, are you ready for this? RNA. We start copying our DNA with actually some RNA. We make a primer out of RNA because polymerase 3 needs a place to start. DNA polymerase 3 cannot start without something already there. And so we add RNA or DNA primase to make a little chunk of RNA, maybe just like 10 to 15 nucleotides of RNA, which is a little bit less high maintenance, a little bit easier to get started. And then DNA polymerase 3 here, can come in and start to bind together those nucleotides that are complementary to our template strand, always in a 5' to 3' direction. It can go continuously on our leading strand. It's going to go away from the replication fork on our lagging strand. And these little fragments, again, are called Okazaki fragments. Each of our Okazaki fragments is going to start with a primer. So I'm going to have a little piece of RNA here, and here, and here, and there used to be one over here. Except we've got another super cool enzyme called DNA Polymerase 1, and it's going to come in and remove that RNA primer, and then fill in that little gap with some DNA instead. It's going to glue it together, except for that one last little 5' to 3' gap that it just can't bind together. And so we have one last enzyme that we're going to talk about. There are more than this. These are just the ones that we're talking about. One last enzyme called DNA ligase. And ligase is going to glue together those last tiny little gaps between those fragments that were left when we pulled out those primers. On the next slide, I have all of these enzymes written down in words. So don't panic if you didn't get all those words quite yet, because here are some descriptions of all those enzymes that we just talked about. Feel free to pause the video, write down whatever you want to write down. Otherwise, we're going to go ahead and wrap up our lecture with a quick look at DNA proofreading. Replication happens crazy fast, and so it is expected that some mistakes will get made. So here we have DNA polymerase 3, gluing together some nucleotides, just chucking along, doing its thing. But here we have a mistake. And interestingly, before DNA polymerase 3 slides all the way away from the DNA that it's replicating, it'll just give it a little bit of a squeeze to make sure that the width of it is just right. And if it notices that the width is either too bulky or too skinny, then this last little chunk of the DNA polymerase 3 will have a little bit of a conformational change. It'll actually remove that wrong nucleotide and put the right one in its place. We think that this mismatch happens 1 in every 10,000 bases or so, which doesn't sound like a lot, but when you think about the fact that our human genome is about 3 trillion nucleotides big, that actually adds up to a lot of mutations, which would be really bad for us. So this proofreading, DNA polymerase checking its own work before it leaves its work, it's kind of fabulous for us. In our class, we're going to watch a super short animation showing how this happens. If you are watching this video and not in my class, you're not going to see that animation when you come to class. I highly recommend that here in YouTube, you search for DNA polymerase Garland Science. They made a beautiful animation showing how the DNA proofreading happens. Highly recommend. It's 2 minutes long. So definitely check that one out. And on that note, my friends, we are done. Objectives accomplished. We defined replication as being that copying of DNA to make exact genetic sequences. We talked about the importance of complementary base pairing. This is how we get the right sequence of DNA to be formed in our new strands. Semiconservative nature of replication. Half of our new DNA is actually old DNA. We talked about functions of enzymes that catalyze those reactions, those condensation reactions that bind together the A's, T's, C's, and G's in our new DNA. We talked about helicase and polymerase in our core content. We talked about some more enzymes in the additional higher level content. We talked about applications of biotechnology based on replication, PCR, polymerase chain reaction, gel electrophoresis, and then DNA profiling. We reviewed the counting of the carbons in the deoxyribose sugar so that we could describe the directionality of DNA replication. 5' to 3'. 5 with the phosphate group is going to get glued onto that 3' carbon corner of the sugar that comes before it. We talked about continuous replication on the leading strand, non-continuous replication on the lagging strand, because we could only go in that 5' to 3' direction, and that leads to the formation of Okazaki fragments that need to get glued together. And we wrapped up with that look of DNA proofreading. DNA polymerase 3 can check its own work to reduce the number of mutations that get added to our DNA during replication. Great work today, my friends.